Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is for 16th February 2021. As all of us know, we are into the revision mode and we are revising the entire syllabus in the form of CBSE sample papers. And until now we have completed one CBSE sample paper. We are reflecting upon sample paper 2. In this, yesterday we discussed up to question number 27, that is section C. Now straight away we go to question number 28. Differentiate between carboxylation and oxygenation. Now carboxylation, whenever I said differentiation questions are there, always you tabulate and explain the answer. So carboxylation one side, oxygenation one side. Under carboxylation, it is the first step of the Kelvin cycle, that is C3 in biosynthetic phase. Under oxygenation of RuBP in the presence of O2 is the first reaction of photorespiration. Carboxylation occurs inside chloroplast. Initial oxygenation occurs inside chloroplast. Second oxidation takes place in peroxisome. This is important. You must note this point. And ribulose 1,5-biphosphate combines with the molecule of a carbon dioxide in the presence of lightly regulated enzyme RuBP carboxylase. Whereas here under oxygenation, photorespiration is initiated in the chloroplast in presence of light only. 2-carboxy-3-coato-arbinatol 1,5-biphosphate is formed. Here of course Rubisco catalyzes oxygenation. It leads to the formation of phosphoglycolate, which is a 2-carbon one. Now question number 29. Name 3 phases of interphase. Give one major event of each phase. Okay, he is asking 3 phases of interphase. So read the question carefully. The 3 main events that occur in interphase are as follows: G1 phase, that is gap 1, post-mitotic and pre-synthetic phase. S phase, synthesizing phase, and G2 phase, gap 2 phase, post-synthetic and pre-mitotic phase. Major events of each phase are: 1. G1 phase is the longest phase during which synthesis of RNA and DNA takes place. Longest phase, G1. S phase is the phase during which chromosomes replicates and prepare themselves for equal distribution. G2 phase is the phase in which synthesis of DNA gets stopped. DNA synthesis stops. However, the formation of RNA continues, takes place. Question 30. What are the benefits of 5-kingdom classification over a 2-kingdom classification? 2-kingdom classification was unable to accommodate certain living beings as they neither had properties of animals nor of plants. Though 2-kingdom classification, it was not possible to include every set of characters in particular kingdom. 5-kingdom classification could accommodate more set of characters. As a result, it was easier to categorize all living beings. For example, we are substantiating. Bacteria can be autotrophic as well as heterotrophic. It was a difficult question to categorize them either under plant or animal. A separate kingdom was most logical choice. Similarly, viruses are in the borderline case between living and non-living. When they don't get host, then they behave like non-living, like COVID-19. Once inside a host, they spring to life. Although the 5-kingdom classification does not have room for virus, but it opened the way for a better and more inclusive system of classification. Hence, 5-kingdom classification is always good. List out, choice is there. List out some living features of viruses. Now here also question, you must be careful, living features, though we consider them as border cases. Viruses, of course, they have DNA and RNA, viral DNA and viral RNA. Most of the viruses have RNAs. Their nucleic acid can undergo mutation. That is the problem we are having now. COVID-19 second strain also started in Europe. So there will be always mutations. Third, they can reproduce only in living host cells. That's why we should not allow them. Fourth, they infect different types of host cells. Of course, nowadays, even COVID-19, we are seeing animals getting it. Okay, recently 2 tigers also died. 2 nucleic acids of viruses are identical in composition to those of other organisms. So, this is how you must answer for 3 mark question. Now, section D is long answer type. There will be 3 questions. Since long answer you have, for every question there is a choice. Each question carries 5 marks, so 15 marks section D contains. What is the question? 31. Amphibians are supposed to be the linking point between aquatic and terrestrial life. Explain. Amphibians are adapted to survive in both aquatic as well as terrestrial life, as research has proved that the life started in water and finally moved to land and air. Aquatic has its own limitations. The area is limited. There is less variety in primary producers, which limits the type of food available to animals living in water. Just imagine, I gave the example. If a mummy prepares breakfast idli every day, you will enjoy for 2-3 days. Afterwards, imagine she prepares idli every day breakfast as your for a month. Then 31st day, what is your fate? So that is the problem. The area is limited and the food is also no variety. Especially in deep sea, limited reach of sunlight doesn't permit variations of certain kinds. Following paragraphs highlights certain features which amphibians developed to adapt in a terrestrial environment. So, emergence of lungs enabled frogs to breathe in air. When they are in water, they breathe through skin. Some aquatic mammals like whales need to come onto surface frequently to breathe in oxygen, which limits the time they can chase a prey underwater. So frogs are having better mechanism to do so. Emergence of forelimbs meant frogs can travel larger distances. Their webbed fingers enable them to swim, while the stronger hind legs are used for better jump. Ease of mobility is helpful from 2 perspectives. One is in getting food and another is in saving from predators. Frogs are amniotic, this is important, amniotic, which is more suitable for aquatic life. But later, animals have switched over to ureotelic and uricotelic mode of excretion. The feeding habit of frogs enables them to survive both on land as well as on water. That's why we say amphibians. While on land, they eat small insects. Presence of nictitating membrane in the eyes allows them to swim underwater. In higher animals like mammals, nictitating membrane becomes, like in our case, vestigial organ, showing their relationship with aquatic life. Now, you have got a choice for this. What is the choice question? Give a comparative account of chordates and non-chordates. Discuss certain characters of chordates that are beneficial from evolutionary point of view. So again, tablet, chordates one side, non-chordates. Notochord is present. Notochord is absent in non-chordates. Central nervous system is ventral in non-chordates, whereas in the case of chordates is dorsal, and they are ventral as well as solid and single and double. Pharynx perforated by gill slits. Gill slits are absent. Heart is ventral. Heart is dorsally present. Post-anal part, that is tail, is present. Post-anal tail is absent. What are the evolution steps of chordates? The presence of notochord is a sign of better development of body architecture, as notochord develops into vertebral column. So it gives more strength and support to the body. In non-chordates, a single nerve ganglion or a couple of nerve ganglia is responsible for all neural control. In chordates, there is a more elaborate system of central nervous system and peripheral nervous system for better control and coordination. Pharyngeal gill slits rule out the need for ingesting large volume of water for food ingestion, and it is an evolutionary step towards terrestrial life. The notochord and post-anal tail help the aquatic animals to swim at faster speed. Development of musculoskeletal system is another means to attain faster speed of locomotion. So 32. What are the different classes of enzymes? Explain any two with the type of reaction they catalyze. Enzymes are divided into 6 classes, each with 4 to 13 subclasses and named accordingly by 4-digit number. We are going to discuss only the main ones because different class of enzymes. Reductases, oxidoreductases or dehydrogenases, enzymes which catalyze redox reactions between two substrates S and S dash. Alcohol dehydrogenase reaction, acetaldehyde plus NADH2, ethyl alcohol plus NAD. Next, transferases, enzymes catalyzing a transfer of group other than hydrogen between a pair of substrate S and S dash, like glucokinase, glucose plus ATP, glucose 6-phosphate plus ADP. Next, hydrolysis, enzymes catalyzing hydrolysis of ester, ether, peptide, glycosidic, carbon-carbon bond, carbon halide bond, phosphorus and nitrogen bonds. All digestive enzymes come under this category. Glucose 6-phosphatase, glucose 6-phosphate plus water, glucose plus phosphoric acid, hydrolysis. Liases, enzymes that catalyze the removal of groups from substrates by mechanisms other than hydrolysis, leaving double bonds. Pyruvic acid carboxylase. Now, pyruvic acid becoming acetaldehyde plus CO2. Isomerase, as the word says, isomer, includes all enzymes catalyzing interconversion of optical, geometric and positional isomers. Example, triose phosphate isomerase. Next, reaction, glyceraldehyde 3-phosphate becoming dihydroxyacetone phosphate isomer. And last, of course, ligases, enzymes catalyzing linking together of two compounds. Enzymes that catalyze joining of CO, CS, CN, PO bonds. Example, acetyl-CoA synthetase reaction, ATP plus acetic acid plus CoA, forming AMP plus phosphate plus acetyl-CoA. This is very important question and the answer is very beautifully given. And now describe the mechanism of enzymatic action. Of course, lock and key. Enzymes act by lowering the activation energy of reactions by creating an environment in which the transition state is stabilized, straining the shape of a substrate by binding the transition state conformation of substrate product molecules. The enzyme distorts the bound substrate into transition state form, thereby reducing the amount of energy required to complete the transition. Second, lowering the energy of the transition state but without distorting the substrate by creating an environment with the opposite charge distribution to that of transition state. Providing an alternative pathway, for example, temporarily reacting with the substrate to form an intermediate enzyme substrate complex, which would be impossible in the absence of enzyme. Reducing the reaction entropy change by bringing substrate together in the correct orientation to reactant. Considering delta H plus alone overlooks this effect. Increase the temperature speed of reaction, thus temperature increase helps the enzymes function and develop the end product even faster. However, if heated too much, enzyme shape deteriorates and only when the temperature comes back to normal does the enzyme regains its original shape. Some enzymes like thermolabile enzymes work best at low temperatures. Then, the catalytic cycle of enzyme action can be described in this. First, substrate binds to the active site of the enzyme, step 1. Step 2, the binding of the substrate induces the enzyme to alter its shape, conformational change, fitting more tightly around the substrate. Active site of enzyme, which in close proximity of the substrate breaks the chemical bonds of the substrate and the new enzyme product complex is formed. The enzyme releases the products of the reaction and a free enzyme is ready to bind to another molecule of the substance and run through catalytic cycle once again. Now we come to the last question. Differentiate between systolic pressure and diastolic pressure. Systolic pressure and diastolic. Remember RD acid, Republic Day. Relaxation, diastole. Here systole opposite. When the blood pressure is highest, when the heart beats, heart muscle contracts, the systolic pressure is generated. Diastolic, when the blood pressure is minimum, it is called diastolic pressure. Diastolic pressure is produced between the beats or at the resting of the heart muscles between the beats. This is the time of refilling the blood. Blood pressure inside the arteries is maximum. Blood pressure inside the arteries is minimum. Systolic stage occurs when the left ventricle gets contracted. Diastolic stage occurs when the left ventricle gets relaxed. Blood vessels contract, blood vessels relaxed. 90 to 120 mmHg in adults, 100 mmHg between 6 to 9 years, 95 mmHg in infants. What about here? Diastolic 60 to 80 millimeter Hg, 65 mmHg 60 to 90 years, and of course 65 mmHg. Of course, nowadays there is a correction here. Even in adults, it can go up to 140. The American as well as British doctors have suggested, particularly cardiologists, that in human beings, particularly adults, it can go up to systolic pressure can be up to 140. Still, they consider it as normal. This is the latest correction that is done. Of course, Indian doctors still they consider 120 as normal. List the main functions of blood in our body. Main functions of blood: oxygen carrier, okay, removal of CO2, transport of food material, transport of other substances, and of course